Hello, dear colleagues. My name is Valeria Timonina, and in this video, I will present to you our poster that's called Heat Shock Protein 90 as a long-term buffer of a species-specific mutational burden. In several works, it was shown that HSP90 can buffer manifestation of deleterious variants. And if we artificially decrease the level of expression of HSP90 by mutations or adding specific inhibitor, these deleterious variants will appear in phenotype. Okay. In this scheme, the balloon represents the buffering activity of HSP90, and as soon as it's big enough, it will hide the needles of the hedgehog that is inside. And if we blow the balloon, that is, we decrease the level of HSP90, we will see the needles. And the needles are, of course, the deleterious variants that now are visible. Also, they were hidden before. And all such works were performed in a scale of several generations of model organisms, and we call it short-term evolutionary scale. In this work, we check if this buffering activity can also play a role in the long-term evolutionary scale. And if this buffering activity of HSP90 does exist in the long-term evolutionary scale, we expect it to be more important in species with low effective population size, such as elephants, because such species have strong genetic drift, which leads to accumulation of high mutation burden. And so these high mutation burdens should be compensated. In the opposite, in species with high effective population size, such as mice, the purifying selection is strong enough to eliminate such deleterious variants from population, and so such species have a low mutation burden, and compensatory role of HSP90 should be less important in such species. Our first expectation was that in species with high effective population size, the level of expression of HSP90 should be lower compared to species with low effective population size. And to check this, we analyzed published transcriptome data for primate species in different tissues. And we saw that in macaque, that has the highest effective population size across primates, the level of expression of HSP90 is lower compared to gorilla, that has a lower effective population size, almost in all tissues, which is in line with our expectation. Then it is known that species with a low effective population size accumulate more non-synonymous substitution compared to species with high effective population size. And to hide all this, the balloon of such species should be bigger. And on this picture, we see that if we regress the NDH ratio, which is the ratio of non-synonymous to synonymous substitution rate, and its approximation of the strength of purifying selection against body mass, that is a reverse approximation of effective population size, we will see the positive slope, and we call this slope relaxation of purifying selection. And taking into account the specific role of HSP90, which should be especially important in species with low effective population size, we expect that the level of relaxation of HSP90 should be 0 or at least less compared to other genes. And to check this, we build a simple linear model of the NDH ratio versus logarithm of generation length, which is another approximation of effective population size. And we saw that indeed HSP90 has a 0 slope, and moreover, it is significantly less compared to the slope of control gene that we chose as the closest one to HSP90 based on structural characteristics and characteristics of evolutionary constraint. Also, we compared HSP90 with a subset of similar genes. We chose 300 genes, and for each gene, we built the same simple linear model and compared the level of relaxation of all these genes. And in this distribution, we see that the level of relaxation of HSP90 is in the bottom of the distribution, and so these results are in line with our expectation. Next, we expect that client proteins of HSP90 should accumulate more slightly deleterious mutations compared to non-clients. If in non-client protein, the proteins that folding is not dependent on the HSP90 activity, and the slightly deleterious variant that disturb the native conformation of the protein will appear. It can lead to elimination of such variants by selection. And if such mutation will appear in client protein, it may be hidden by the buffering activity of HSP90, and the native conformation will be restored, and so it can lead to accumulation of such mutations. To check this, we chose the client proteins of HSP90, and for each client we chose the control gene, the closest control gene, based on structural characteristics and characteristics of evolutionary constraint. And for each gene in these two subsets, we built the same simple linear model and compared the level of relaxation in two groups. And in this picture, you can see the marginally significant trend that HSP90 clients has a higher level of relaxation of purifying selection compared to non-client proteins. So next, we switch our focus from comparative species analysis to analysis of human population. And first, we collected data about metrics of evolutionary constraint as well as the structure features of more than 11,000 human protein coding genes. We performed the principal component analysis, taking into account all these characteristics. And we saw the distribution of client proteins in the space of two first principal components. And we see that such metrics as selection coefficient of heterozygosity, probability of being loss of function intolerant, residual variation in intolerance score, they all reflect the evolutionary constraint and they contribute more to the first principal component. And in the second principal component, the most contribution is from the metrics of structure characteristics of genes. So we can say that PC1 approximates the level of evolutionary constraint and that clients are shifted to the side of less constrained genes. Next, we expect that loss of expression of HSP90 should be more deleterious than gain of its expression. And so we expect that HSP90 should have a deficit of regulatory variants that decrease its level of expression. And to check this, we analyzed data about these regulatory variants from GTX. And we calculated the proportion of variant that decrease the expression level of genes for more than 18,000 genes. And we see on this distribution of proportion of loss of expression variance that HSP90 is in the bottom of this distribution. That is, it indeed has the deficit of loss of expression variance. Next, we compared the level of expression of HSP90 in different human tissues, and we divided the tissues in 3 groups according to their turnover rate. And we saw that the highest level of expression of HSP90 is in the slow replicating tissues. And we can't draw a strict parallel between slow replicating tissues and species with a low effective population size, but we can speculate about it. And in any case, this observation is definitely worth more research. And to sum up, we observed several lines of evidence in both comparative species and human population levels that HSP90 can work as a buffer of mutational burden. And in the end, I would like to thank my colleagues that contributed to this work. Thank you for your attention, and don't hesitate to contact me if you have any questions.